Because despite the variation, there are many different kinds of signaling pathways. All signaling pathways have to have a couple critical components. And they have to have certain features about them. The rest of it is a whole bunch of different details. And yes, you're going to get a whole bunch of different details. And you'll end up seeing these things on exams and stuff like that. Okay, but we need, the elements we need for a signaling pathway include, first of all, we need to have a receptor. We got to have a sense organ. Using the eraser analogy again, if she was blind and I threw the eraser at her, she wouldn't be able to respond. She can't pick the signal up. Or put a blindfold around. When will you get hit? Okay. Yep. All right. She has to have the sense organ to detect that change in the environment, like the flying eraser. Okay, so first thing we need some, oh, I hate this. One day I'm going to slip, it's going to end up all over the internet. 4-letter words describing this cheap chalk that they give us. Okay, let's see, we got, watch this all shattered too. They used to make good chalk one day that would actually hold together. Now they gave us L-cheap stuff. But anyway, okay, first thing we need a receptor protein. That's the sense organ.
This is what we need for signaling pathway?
Yeah, these are going to be the 3 key elements of a signaling pathway. And all signaling pathways are going to have something like that. It's just what it is varies from pathway to pathway. So we need some kind of receptor protein. That, in effect, is the sense organ. Now, a receptor protein has to have several qualities about it. One of them, whatever the signaling agent is, does not actually get taken inside the cell. If it's a molecule, like a, well, steroid hormones are an exception, if it's a signaling molecule, that molecule stays on the outside of the cell. The cell does not pick the thing up and take it inside. Just like, here again, if I threw the eraser at her, again, a kid with erasers all the time. If I threw the eraser at her, the eraser is going to be on the outside of her. It's not going to go inside her. Unless I have Superman to throw it 1,000 miles an hour or something, but it's not going to go inside her. It's going to be on the outside. Okay. So, that means the receptor has to be able to detect something on the outside, whether it's a molecule, pH, any of a number of things. Light or what have you, any of a number of things. But that receptor has to be able to communicate with the inside of the cell, the signaling pathway that's inside the cytoplasm. So what that means, the receptor has to completely span through the membrane. Part of it on the outside, part of it on the inside, and the waste is buried in this nice, greasy, hydrophobic membrane. So it must span through the membrane. Like so. Okay. Now, the second thing is this. Let's suppose, well, let's just, since this is mostly used, let's suppose we're dealing with some kind of molecule. This receptor protein is designed to bind that molecule. So here's a molecule. In fact, we will call it, and we use the generic term, we'll call it a ligand. Okay, and the term ligand is a generic term. It's kind of like the term substrate referring to something that an enzyme binds and catalyzes. A ligand is a molecule or molecules that bind a particular receptor. So each receptor has its specific ligand or ligands. For instance, the protein insulin produced in the islet cells in the pancreas. It's a little small protein. Cells have a receptor called the insulin receptor. And that protein binds insulin. and then it initiates a signaling pathway that regulates cell metabolism and a whole bunch of other things. Okay, so you could say that insulin is the ligand of the insulin receptor. Question?
Are all receptors specific, or are there some that are some specific, some broad?
Some are broader than others. Most are fairly specific, but you might have several molecules instead of one, and others are extremely specific. And not only that, the same molecule can also bind to a family of receptors. So I'll give you an example, neurotransmitters. Now they bind, that sends signals from across the synapse from one nerve cell to the next. Neurotransmitters will bind these neurotransmitter receptors, which generally open up or themselves are ion channels and let ions go in and out of the nerve cells. There are many neurotransmitter receptors where there's several kinds that all bind the same neurotransmitter and have somewhat different effects. And that's kind of like the next hope in pharmacological drugs that affect brain function is that right now about the best they can get are things that bind a whole class of receptors, like those antidepressants called the SSRIs, Prozac and Lexapro and those kind of things. They bind something called the serotonin receptor. Serotonin is one of the neurotransmitters. But there are several different types of serotonin receptor, and they each kind of have different effects in the central nervous system. It would be nice to get something that would bind to one type and not to any of the others. Then you would have a much more specific drug with fewer side effects. So here again, so you can have multiple receptors for the same ligand in many cases. Did that help? Okay, all right, so receptor has to span the membrane, but one of the most important things about receptors, here we have the ligand binding the receptor. How does the inside of the cell know that an event on the outside has taken place? Well, here's the key to how receptors work. The receptor has to change its tertiary structure, or quaternary structure if it's part of a complex. It has to alter its tertiary structure. And it has to alter it in such a way that the inside part has undergone a structural change. So let's put that down. Okay, so must alter its tertiary structure in response to ligand binding. So, now we have the ligand bound here, and we alter the structure, and I represent it by this. Now, the inside of the cell sees this part of the protein as looking different than it did before. Because this is what happens when you bind the ligand. When you don't bind the ligand, it has a different shape or different structure. So that's another thing. Now that's true of all these kinds of receptors, not just ones that bind molecules. We have many other kinds of receptors that are going to respond to different things. For instance, we have light receptors. Think of the retina. What happens? Light absorbs a molecule called retinol, which is embedded in these proteins, the rhodopsins as they're called, inside the nerve cell membrane, inside the retinal cell membrane. When that molecule absorbs light, it undergoes a structural change. It bends, and that causes the entire protein to bend. So that initiates a signaling event that ultimately leads to ions rushing in and out of the cells in the retina, and that fires off a nerve impulse, and the nerves are making close contact with those things. So, in this case, absorption of light by these different photoreceptor cells. That causes a change in the receptor and initiates a signaling pathway that ultimately leads to vision. We have heat and cold receptors. They alter their tertiary structure when the temperature goes up to a certain point or goes down to a certain point, and then you feel the sensations of heat and cold. Now, an interesting little sidelight on that. Turns out, they found out a few years ago, the reason that hot chilies taste hot is that capsaicin. That's the active ingredient in chilies, and that's what they also put in those boyfriend sprayers, yeah, that's what I call them, boyfriend sprayers, you know, when they carry around to keep their partners in line, but the pepper spray thing. And,
I thought you were talking about like perfume.
excuse me?
I thought you were talking about perfume.
Oh, no, I was talking about, I think it outlined, this keeps in line, but the pepper spray, you know, it also contains that. Well, the stuff tastes hot because it just so happens that that molecule binds to the temperature receptors in the cells and causes the same structural change that heating those receptors up also causes. So the inside of the cell does not know that it's a chemical. Rather than dangerously high temperatures, it responds the same way. So if you do something stupid like eat a raw habanero, trust me, do not try that. You will regret it, okay? And your mouth is going to feel like somebody poured boiling water into it because you're stimulating the temperature receptors, in this case, by a particular plant product. And the two work synergistically too. I remember one time I was chopping up some banana chilies, which are really weird because these banana chilies, they can taste, some of them are real mild, they taste almost like a green pepper, and others are screaming hot. They will make jalapeno look mild. Okay, so you never know what you're getting when you pick them up at the supermarket. Are they really hot, or are they really mild, or somewhere in between? So I'm cutting these things up, and you know, I feel a little bit of burning sensation in my hand and stuff, a little warmth and what have you. Okay, that's to be expected, right? Then I take a shower, and nice warm shower, and feels great, except for my hands, it felt like I was sticking them in scalding hot water. So the effects of the chemical, which was already stimulating the receptors, and slightly warm water added together, and it felt like I was putting my hands in scalding hot water. Even though the rest of my body felt like it was a typical warm shower, my hands felt like it was unpleasantly hot. So that happens to be a case of a molecule, plant product, producing the stuff that happens to stimulate the same heat receptors. And generally, most animals will not, after an experience eating a hot chili, will not eat. Will stay away from that plant from now on. That's a good way of preventing predation. Except for people like Asians, Latinos, and many Africans who like very hot food. Okay, well, at any rate, so that's a key point. The receptor has to change its tertiary structure in response to ligand binding. Now, second thing we have to have, we've got to have some kind of molecule that is going to bind to the receptor in one of the two states, either the empty state or more likely the state that has the ligand-bound or occupied state. Now let's just call that molecule for a time being an intermediary molecule. We won't say what it is, but, and it varies. Okay, these intermediary molecules, they have to recognize the difference, the structural difference between an empty or vacant receptor and an occupied or ligand-bound receptor. And then it has to be able to bind to one of those two states, but not the other. Now, most of the time it means we bind to the occupied receptor and ignore the empty or vacant one. But that is not always the case. But you have to be able to tell the difference between an empty and a ligand-bound receptor, and you have to bind to one of the two. Okay, so that's one thing it has to do, but that's not all. Once that happens, once we bind to the receptor, this intermediary molecule has to undergo its own structural change. And that structural change is then going to be recognized by the rest of the pathway. So we'll say must Okay, so second critical point, that intermediary has to undergo a structural change when it binds the receptor. And that's ultimately going to lead to activation of the rest of the thing. So over here, if we have an intermediary, and I'll just call it I, here's our intermediary, whatever it is. It binds to, in this case, the occupied receptor, and then it undergoes a structural change. In response to that binding, and quite frequently it falls off the receptor. Not always, but quite frequently that change allows it to release from the receptor and then go off into the cytoplasm and do whatever it does. So that's step 2. Now, often these intermediaries are the molecules known as G proteins. They're commonly used in signaling pathways. Now, not all signaling pathways by any stretch of imagination use G proteins. Many signaling pathways do, and we'll look at these guys separately and later on. Many signaling pathways do indeed use G proteins, but many of them also do not. They'll have other things as intermediaries, and we'll see examples of both. Okay, so that's step 2. Now, let's go to step 3. I'm going to erase this here. We've activated the intermediary. Now that, so far what we've done here is one molecule, one ligand, is bound to one receptor, and it's activated one or maybe a handful of these intermediary molecules. Okay? That's just a handful of molecules. That is not going to do too much to a cell that contains, millions or tens of millions or billions of individual protein molecules and what have you. You just have a couple of molecules here. It's not going to affect much. So now what we need is to take that little signal here, those few intermediary molecules we've activated, and now amplify that so that now we affect hundreds or thousands or tens of thousands of molecules, enough in that cell to give us a final cellular response. So the third thing we need is some kind of, we could call amplification steps. And that's generally done by these intermediaries. These intermediaries, like G proteins, they will bind to things and various things that lead to the amplification. That can include things like activation of protein kinases. These are the regulatory enzymes. God, I hope you guys know this. These are the regulatory enzymes that phosphorylate covalently attached phosphates to other proteins in order to regulate them. They can activate other types of enzymes. They can activate, directly or indirectly, transcription factors. In other words, regulating gene expression. They can activate membrane transport. Those of you who remember cell probably realize that calcium ions inside a cell can do all kinds of things. There are many calcium-regulated proteins in cells. Now imagine, and that's why cells keep internal calcium levels down to less than 1 micromolar. Now imagine what would happen if we opened up calcium channels and let calcium ions rush in. That would certainly give us a dramatic effect. As a matter of fact, that's one of the key events in the formation of the fertilization membrane, is calcium ions rush into the cell and cause secretion of these so-called cortical vesicles that end up turning the vitelline membrane into the fertilization membrane. And the proteases within them are the very things that we are going to inhibit today in lab. Okay, so clearly you can have all kinds of different things. Now many times you're going to have a multi-step amplification. Maybe you activate a kinase that in turn activates other kinases. So now you get from a little small signal, now you get a really big signal. Now let me give you an analogy. How many people have stereo systems at home? I'm not talking MP3 players or anything, I'm talking a real stereo system. Okay, a few of us, good, I'm not getting that old yet. Okay, all right, now the thing is, I used to be a holy grail when you went off to college back when I was in there. You got yourself a really nice, decent stereo system. Set you back about $1,000, but you got a good one. Well, anyway, how does a stereo system work? Think of this. You're getting, you're putting on a CD, okay, or a magnetic tape, remember those? Or God forbid, a vinyl album with little squiggly lines in it? Things your parents and grandparents might still have. Okay, well what's going on? Whatever kind of signal you're getting from is going to be measured in milliwatts. It's a very tiny electrical signal that you get from wiggling a needle through around a whole bunch of squiggly lines in the vinyl, or reading the tiny little magnetic field differences in the tape, or shining a laser through the holes in a CD. However you do it, you're going to get a little tiny initial signal in the milliwatt range. That's not going to do anything. So what you do is then you take it into the receiver, that little signal, and they have what they call a preamplifier. It takes that signal, keeps the same waveform of it, but it makes it much stronger. So now you take that signal of a couple milliwatts up into several hundred milliwatts. That's enough to power a set of stereo headphones that you plug in. That's why you have a separate plug for those things in a typical stereo system. It's nowhere near enough to power a set of speakers, unless you got some really tiny little speakers. Okay, and then you take that signal and do a second amplification, what they call the power amplifier. And that takes that signal and ups it from several hundred milliwatts up to 50 or 100 or 200, however much, however powerful and however much money you got to buy the system. And those tens to hundreds of watts, that signal is enough to drive your speakers. And if it's a real powerful system, it's enough to get your neighbors to call the cops on you. Okay, so in this case, your typical stereo system, you have a two-stage amplification. A weak initial signal that can't do much of anything, but you amplify enough to drive some headphones, and then you run it and amplify it a second time enough to drive your speakers. Okay, that's how stereos work. Okay, now, signaling pathways are often the same thing, where you have not just one amplification step, but you have more than one, and that means a little tiny signal of a few ligands binding to a few receptors ultimately leads to some kind of response that's affecting thousands of molecules in the cell. You may activate a battery of transcription factors, and they're going to be turning on genes or turning off genes, up, down, left, right, and center. That can lead to massive changes in cell differentiation. You can regulate all kinds of membrane transport channels and active transport and what have you. That's going to cause all kinds of major changes in cells. So the amplification step, or steps, is very important. And because many times you have more than one amplification step, it means ultimately these initial signaling molecules are biologically effective at extremely low concentrations. For instance, the cytokine injection I get every month after each of my rounds, that is 0.6 milligrams. Yet it's enough to stimulate the production of certain types of white blood cells throughout my entire body. That's not also cost $5,000 a pop. I'm sure it's government paying you for it, but that kind of thing. A fraction of a milligram can make of this signaling molecule, it's a cytokine, can make dramatic changes in your immune system. It can stimulate the production of enormous numbers of certain types of white blood cells. So it tells you something. These signaling molecules can often be very effective at extremely low concentrations because of those amplification stages. Okay, so those are the major features we see that are common to signaling pathways. Now, when we go through specific pathways, you might want to keep that in mind and say, oh, well, here's the receptor, and okay, we're using this or that as an intermediary, and finally, here's the amplification step or steps. Okay, does anybody have questions on that aspect?